Hello students, welcome to E-Patasala. I am K. Muralidhar from South Asian University. I am an honorary professor there. Today we are going to talk living processes about nutrition and energy, and this forms part of the paper on introductory biology. What do we learn from this paper? The learning outcomes as listed here. At the end of learning this module, one would understand that there are autotrophs and heterotrophs among living organisms. Realize that only green plants are autotrophs, that is, make their own food. Know that there is a huge diversity among heterotrophs with regard to nutritional requirements. Appreciate the fact that food that is taken in serves two purposes. One, it is a source of metabolic energy, and two, in the case of higher animals, it also serves as a source of essential vitamins, amino acids and trace elements. Plants also require trace elements. Understand that metabolic energy in the form of chemical bond energy is generated, stored and utilized by all living organisms as a ceaseless activity till death. Realize that the storage form of energy is different in different organisms. That is, it is glycogen in animals, starch in plants, polymetaphosphate in certain microbes, etc. Appreciate the fact that the energy currency, however, is same in all organisms, and that is in the form of nucleotides like ATP or GTP. Realize that organisms perform three types of work, that is osmotic, mechanical and biosynthetic work. All of these require energy and hence coupled to ATP hydrolysis. Let us see what this lesson is about. This lesson is to live and generate energy, living organisms need food. Food serves not only as a source of energy, but also in some cases, like most animals and plants, as a source of essential components which cannot be manufactured by that group of organisms. Animals require vitamins, amino acids, unsaturated fatty acids and trace elements like iron, magnesium, zinc, etc. Living organisms are divided into two major groups: autotrophs, for example green plants, and heterotrophs, consumers of all types, depending on whether glucose can be manufactured by the organism or not. Plants which have the green pigment chlorophyll, usually in leaves but sometimes in other parts also, synthesize it glucose oblique starch with the help of sunlight, water from soil and carbon dioxide from air in a physiological process called photosynthesis. Heterotrophs, on the other hand, exhibit a wide range of dietary requirements. Escherichia coli, a bacterium, for example, requires a carbon source like glucose or anything that can be converted into glucose like lactose. It requires a nitrogen source, an ammonium salt usually, and a couple of inorganic cations and anions. A human being requires, on the other hand, carbohydrates, proteins, fat, unsaturated fatty acids, vitamins, certain amino acids like L-arginine, L-leucine, certain elements like iron, magnesium, calcium, copper, selenium, etc. Evolutionarily speaking, the more specific the dietary requirement, the more vulnerable is that species to threat and extinction. One should cultivate eating a broad range of foods. Insects, on the other hand, can make the vitamins but cannot synthesize cholesterol, which has to be provided in the diet. While herbivores can consume plant material and get their nutritional requirements, carnivores may become deficient in due course of time. Green plants are autotrophs. Higher plants for example flowering plants, contain the green pigment chlorophyll, and with the help of sunlight utilize atmospheric carbon dioxide and water taken up by roots to synthesize glucose. This slide deals or tries to show you picture of a green leaf and what happens inside the cells of this green leaf. In a previous lesson, we learned that living organisms are essentially energy machines. They produce energy, they store energy, and they utilize energy for performing different types of work. On that basis, there are two types of organisms. Those who are called autotrophs, green plants are examples for this. These organisms can manufacture their food essentially by using atmospheric carbon dioxide and water. However, a large majority of organisms are heterotrophs, animals included in them, in which case the food has to be supplied from outside as they cannot manufacture the primary food themselves. In this slide, you will notice that the leaf structure is given, and this leaf has cells in which there is a pigment called chlorophyll, of which you must have studied in other places, and this is a machine which captures sunlight of appropriate wavelength, and with the help of that energy can synthesize glucose from carbon dioxide and water. In the primary reaction, which is a light reaction, there is a splitting of water, generation of reducing power, and in the second group of reactions called dark reactions, this energy is utilized to reduce and to manufacture so-called glucose from carbon dioxide and water. This ability to prepare their own food from inorganic compounds like carbon dioxide, water, and with the help of sunlight or solar energy resides in these green plants, especially in the green parts, which are green because of the possession of a pigment called chlorophyll. Here is in this slide shown a slide which essentially depicts structure of a chloroplast, otherwise called as plastids, in which these pigments are organized. What you see, therefore, is the structure of a cut section of a chloroplast or a plastid. These pigments, chlorophyll is one of them and the primary important, they are all arranged in majority of plants as two systems, photosystem 1, photosystem 2, but in some, one of them will be absent. Now, these photosystems divide the job of photosynthesis first in the form of a light reaction, followed by in the form of dark reaction. And essentially, the energy of this is coming from sunlight, but this sunlight is captured by the chlorophyll and other pigments, and thereby water is split into oxygen, and some of the electrons will be raised to a high reducing power, and then by circulation of this reducing power through a series of electron carriers or respiratory pigments, finally carbon dioxide and water will be converted into glucose. The first reaction will be fixation of carbon dioxide using an enzyme ribulose 1,6,1,5 bisphosphate carboxylase. So the primary carbon dioxide is fixed on this ribulose bisphosphate, and the intermediate is split into a 3-carbon compound. Subsequently, in what is popularly called as a dark phase of photosynthesis, a series of reactions occur which is also called as a pentose phosphate cycle, in which case we get ultimately glucose by using a lot of pentoses and interconverting them. Two enzymes are very important in this, one which is known as aldotransferase, another which is known as a ketotransferase. This slide talks about the second major group of organisms whom we labeled as heterotrophs, which simply means these are organisms who have to depend upon the autotrophs as a source of food. These are the primary consumers. So if you now look at how they take this food and convert into energy, first of all we should realize that the diet or food that we take contains many components and serves as a source of energy, and the major components which will give us energy are the carbohydrates, fats and proteins. As depicted in this table, each one of them per gram yields certain fixed amount of energy given in the form of kilocalories. As indicated here, fat gives us highest amount of energy, 9 kilocalories per gram, whereas carbohydrates and proteins give us 4 kilocalories per gram each. Subsequently, this diet or food is taken in and undergoes what we know as different phases of metabolism, which in physiology, the phase 1 metabolism is also called as digestion. In biochemistry, this is continued as intracellular phase 2 and phase 3 metabolism. As we pointed out, heterotrophs take their food ingestion and then this is digested in gastrointestinal tract in higher organisms or a similar part of the body in lower organisms. This in biochemistry is also called as phase 1 metabolism. In phase 1 metabolism, the macromolecules are present in the food like proteins, fat, nucleic acids, polysaccharides. They are broken down into their monomeric components. As indicated in this slide, proteins, they are digested in our gut with the use of enzymes called proteases, and the products are the building blocks called amino acids. Similarly, fats are degraded by lipases and phospholipases into their monomeric building blocks called glycerol and fatty acids. Similarly, nucleic acids are broken down into their building blocks called nucleotides. Similarly, but what is not indicated here, polysaccharides like starch, glycogen, any other thing is broken down into their monomeric units, which are monosaccharides or sugar units. What we should remember here is one of the polysaccharides which is present in plants called cellulose can be digested only by certain ruminants, that too with the help of resident bacteria. Otherwise, all the heterotrophs are incapable of digesting cellulose into and converting it into glucose units. Let us understand what are these products of phase 2 metabolism. We have already learned that the food that goes inside our body is digested and assimilated, and this in biochemical terms occurs in 3 phases: phase 1, phase 2, phase 3. At the end of phase 1 metabolism, all these macromolecules are degraded and converted into their monomeric building units. Like proteins become amino acids and polysaccharides become monosaccharides. At the end of phase 2 metabolism, which is always intracellular, the following 6 compounds are invariably formed in all organisms: glycerol, which is a trihydroxypropane; pyruvic acid, which you come across at the end of glycolysis; acetyl-CoA, which is a product obtained after pyruvic acid undergoes oxidative decarboxylation catalyzed by an enzyme pyruvate dehydrogenase; succinic acid and fumaric acid, which are also products of phase 2 metabolism which can directly enter TCA cycle; oxaloacetic acid, a key catalytic component of TCA cycle or Krebs cycle, and alpha-ketogluteric acid, a transaminated product from glutamic acid, are the key 6 compounds. What is interesting here to note that independent of what you eat, the variety of foods that we take, only these 6 compounds are formed at the end of phase 2 metabolism. As we said, at the end of phase 1 metabolism, the products of digestion, as they are called, are essentially building blocks like amino acids, monosaccharides, nucleotides, fatty acids, glycerol and others. Now they enter through portal system into liver, and then the phase 2 metabolism starts. At the end of the phase 2 metabolism, all the monomeric building blocks are broken down into only total of 6 chemical compounds, which we will show later. In this slide, what you are seeing is a metabolic pathway popularly called as glycolysis, in which one of the products of phase 1 metabolism, a product derived from polysaccharides, say glucose, is broken down in a series of 10 metabolic reactions, which are chemical reactions catalyzed by specific enzymes, and the product of this is pyruvic acid. These are 10 steps. However, slight modification occur in different types of organisms, where instead of pyruvic acid, you may get lactic acid, you may get alcohol, you may get organic solvents, you may get antibiotics, you may get other things. This is the primary and the most important metabolic pathway common to all living organisms, unicellular and multicellular. At the end of phase 2 metabolism, as it is called in biochemistry, all these macromolecular components are broken down not only into monomeric building blocks, they are further broken down, and here almost we notice only 6 compounds are generated. That means independent of the diversity of food that we take, independent of the chemical diversity that exists in the diets that we take, ultimately you get only 6 chemicals in all living organisms at the end of phase 2 metabolism. Now each one of them will ultimately be burnt up in phase 3 metabolism, which is depicted here in this slide, and this is known as a circular metabolic pathway. It has got a name, tricarboxylic acid cycle or Krebs cycle, in honor of Sir Hans Krebs, who got a Nobel Prize based on his work on this cycle. In this cycle, you notice that acetyl-CoA, one of the products of phase 2 metabolism from fat as well as from pyruvic acid of so-called carbohydrate metabolism, this enters in this cycle by conjugating to oxalic acid, and you get a primary product, citric acid. Subsequently, in a series of biochemical reactions catalyzed by wonderful enzymes, citric acid becomes isocitric acid, which becomes alpha-ketogluteric acid, which becomes succinyl-CoA, which becomes succinic acid, fumaric acid, malic acid, and back to oxaloacetic acid. And therefore, the second cycle will start. In this process, therefore, acetyl-CoA will be completely burnt out into, again, inorganic components like carbon dioxide and water, and you will notice in this cycle at specific steps carbon dioxide is released. Now this, therefore, the primary result or concept behind so-called phase 1, phase 2, phase 3 metabolism coupled to electron transport in mitochondria is the generation of energy currency. This energy is utilized immediately or stored in certain chemical structures for future use. The whole philosophy of living process is described as utilization of energy, stored energy, in the form of glycogen or starch or polymetaphosphate, depending on the organism, is used to perform work. In the higher organisms like higher vertebrates, this essentially, which contain a complex system called circulatory system, the heart essentially pumps the blood, and the blood carries these nutrients throughout the tissues to all tissues and where the burning takes place, and that is what is shown here. However, the energy that ultimately is produced is called upon to perform work. All living organisms perform 3 types of work. From our understanding of physical principles, work cannot be performed without energy, and therefore this energy or metabolic energy generated by living organisms in the so-called catabolic limb of metabolism is used to perform work. What type of work that living organisms do? One is osmotic work. Osmotic work essentially means you push chemicals and other molecules across a cell membrane but against a constant concentration gradient. Here is one example indicated where what we call membrane transport results in transport of molecules from one side of the membrane outside into the cell. Now where it is only a concentration gradient, it will go through diffusion, which does not require energy, but if it is something against concentration gradient, it has to be pushed, and for that this reaction or transport will be coupled to energy utilization. Usually ATP is used, as is indicated here, to push either glucose or sodium or some other thing. Living organisms from unicellular to multicellular highest organisms also perform a major type of work, which is the second type of work, which is called mechanical work. In higher animals, like the example given here, we have what we call as the muscular system and the skeletal system. Combined, these two skeletal-muscular system enables us to perform mechanical work, as we do when you are lifting a bucket of water, or you are pushing a cricket ball or bowling to a batsman, or you are essentially hitting a ball, football or cricket ball. Wherever there is a displacement in space, you call it a mechanical work. This coupling of energy to mechanical work is mediated by muscle tissue. In the structure of this muscle essentially is given in this, it has series of cells, and these cells, which are also not circular but elliptic and with fibrous ends, they undergo through connections contraction. At the molecular level, their components called actin and myosin fibers slide against each other, and that results in what we call as muscle contraction, and as a result of which the skeleton that is associated with this moves from one place to place, and that results in performance of mechanical work. This slide depicts essentially the structure of muscle. As we understand by now that living organisms produce energy, store them, and then use them to perform three types of work. We have already talked about osmotic work. Then we talked a little bit about so-called mechanical work through our skeletal-muscular system. Here is the structure of our muscle. Each muscle contains, if you divide it, it will has muscle fibers, and then this, if you go further down, it comes to molecular level. Essentially these are actomyosin filaments or protein molecules which slide against each other and give you at the gross level an indication that muscle is contracting. So that's how we perform mechanical work, because this muscle is attached to skeleton, and therefore when the skeleton part like a hand or leg move, we get the impression that we are performing work. Therefore, living organisms perform three types of work: osmotic work, mechanical work, and the third type of work, which is not indicated here, is what we call biosynthetic work. We mentioned that biology is a science of living organisms, and living organisms include both their structures and functions. Structures we have already seen, how these organisms look like, what are their names. Functions are essentially described in two parts: physiology and behavior. And if you look at all these functions, essentially it answers one question: what do organisms do? And therefore, the third major thing that they do is growth. Growth essentially means addition of biomass, which in molecular terms means biosynthesis of all the big molecules from small molecules. Therefore, this requires energy, and hence we say, if you summarize whole biology as what living organisms do, they have structures, they have functions, and the function exhibited by them includes growth, reproduction, memory, defense, and other things, some of which details we have already talked in other lessons. Therefore, biosynthetic work, mechanical work, and osmotic work are the three major types of work performed by living organisms throughout the time when they are called living. The day we are dead, dead bodies don't do voluntarily work, and therefore death is an equilibrium as per our definition. Now, therefore, ability to work is a characteristic feature of living organisms, and that is the most important philosophy guiding understanding biology. Biological systems, through their structures, they interact with each other and also perform a lot of functions. growth is one of the most important functions. Similarly, reproduction, and all these are requiring energy. So, students, let us now summarize what we have learned from this lesson. We have learned that organisms are divided into two groups. Those that can make their own food are called autotrophs, for example, green plants. And those who get their food by feeding on others, these are called heterotrophs, for example, all animals. Heterotrophs represent a wide range of dietary preferences. Living is the summation of all the physiological and behavioral processes. These require energy. Food is the source of energy. Energy for living organisms is mostly metabolic energy in the form of high energy chemical compounds. Food is complex, and when it is digested is essentially broken into simpler structures, thus releasing energy. Part of this is trapped as bond energy in the form of, say, ATP. The efficiency of conversion is close to 40%, and this is more than any man-made machine. This energy enables organisms to perform their osmotic, mechanical, and biosynthetic work. All physiological processes like respiration, reproduction, growth, locomotion, transport, development, and even behavior are different types of work requiring energy. All aerobic organisms utilize oxygen as the terminal electron acceptor, whereby oxygen is reduced to water or partially in the form of reactive oxygen free radicals. Anaerobic organisms use a wide range of chemicals other than oxygen as electron acceptor, for example, hydrogen sulfide and others. Thank you.